See? So this I'm going to tell you something about, well, give you an introduction into virology. And I told you last week about bacteria and how there's many different bacteria on our planet, much more than there's humans. Well, viruses are even more, well, found more frequently. The latest estimates on how many viruses there are on the planet is about 10 to the 30th. Just to give you an idea about how much that is, if you would put all the viruses in a line and stack them up, then we would have a line as long as 200 light years. So there's a lot of viruses, and they are found in pretty much every place on Earth. In animals, in humans, in waters, in hot springs, in plants. Basically every organism that we have can be infected by viruses. So viruses were first seen by this Dutch botanist, and the virus that he found is this virus. He wasn't able to show it as nicely as we see here, but this is what we call tobacco mosaic virus, and you can see here why the virus got that name. This is a tobacco leaf, and this is the leaf when it's infected by the virus, and you see that it gets a mosaic pattern. So this was the first virus that was found. And just to give you an idea again about the size, viruses are on average about 50 to 500 nanometers long, and you have some very small ones, but they are usually the ones that infect bacteria, but the viruses that infect humans are usually something bigger, like this size, smallpox virus. And if you put that in perspective, this is an E. coli bacteria that we talked about last week, and then you see the E. coli and the viruses here in relation to a red blood cell, so they are rather tiny. So this is the machine that can be used to visualize viruses, an electron microscope, and you can get images like this. So there's no other way that we can see viruses, neither by light microscope and of course not by naked eye. So viruses are rather unique, and that's why we're discussing them separately, not together with the other microorganisms like the fungi, the bacteria and the parasites that we discussed last week. And basically you can, the reason why they're quite different is that you could consider them not to be alive. They can only replicate inside a host cell, and they do that actually in a very different way. There is a large variation in replication mechanisms. And the other thing that makes them quite unique is that they are very creative in evading the immune system. And also, you could basically say that they always cause disease. I mean, we talked about last week about commensal microorganisms, and commensals, commensal meaning that one benefits and the other one is at least certainly not harmed. Well, since viruses have to replicate in a host cell, they are pretty much by default always doing some damage. So what are some general characteristics of viruses? Well, like I said, they have to grow inside a cell, so they are obligate intracellular microbes. Their genome can also be different, whereas eukaryotes and also the fungi and bacteria always have a DNA genome. Viruses can have both a DNA or an RNA genome, and it can be double-stranded or single-stranded. Basically, around this genome there's a code, mainly consisting of proteins, and this could be structural and non-structural proteins. That is basically what a virus is. So it's a genome surrounded by a code, and the code can be more or less complex. So, like I said, the least complex codes just are a protein code, which we call a capsid. Some of the viruses also have a lipid and carbohydrate envelope around this. So they must replicate in a living cell. And the replication mechanisms vary, and we will get back to that. And so the replication effect, so the living inside the cell, together with the immune response, is why they are always considered to be pathogens. So here you see some overview of virus particles, and there's many different ones, but I just gave you two examples here. This is a hepatitis diagram of a hepatitis A. You see here the RNA genome, and you see then the protein code around it. So very, very simple virus. And this is a hepatitis B. Again, you see here the hepatitis B genome. In this case, this is a DNA. Then you have a capsid, just like the capsid here. That is the green one here. So you have a capsid around it, but hepatitis B on top of the capsid also has an envelope, and that is depicted here in blue. And around on this envelope you see viral glycoproteins, and those are used as receptors, for example, for adherence. Yeah, this is a polymerase, yes. So here you see the viruses, how they are classified. Generally, the virus classification is in two groups. They are based on their genome, so you can divide them in RNA viruses and DNA viruses. The second grade of classification is usually based on whether they have an envelope or not. And you see here the different virus structures. Now, the ones that we are discussing in this block are these. So we're discussing noro and rotavirus in the next case, and in this case of today we're focusing on the hepatitis viruses, and mainly on hepatitis A, hepatitis B and hepatitis C. And you see that between the three hepatitis viruses they're quite different. So two have an RNA genome, one without an envelope, one with an envelope, and one is a DNA virus with an envelope. So I told you that virus replication is rather different, and well, the goal of the replication is always the same though. It's make progeny, and they replicate inside the host cell, and usually this is quite a nice orderly series of events in which this happen, but the details differ quite extensively. So what I want to do today is to give you an overview or sort of general layout of this replication that you can remember, and if you have this general layout, you can, just by knowing what kind of virus we're talking about, whether it's a DNA or an RNA virus or one with an envelope or without an envelope, you should have an educated guess and be able to see how replication most likely will take place. Now, I won't go into this in detail, but what we do expect is that you know the general processes such as DNA replication, translation, transcription. Those are things that you should know from the human cells. So I won't go into that unless someone has specific questions. No? Okay. So how does virus replication work? So you see here that we have a susceptible host or tissue, and the virus comes in contact with this host or tissue, and the first thing that it needs to do, and this is similar to the infection with bacteria, is that they always need to attach. So if there's no attachment, they cannot start to cause an infection. So they attach themselves, and then they have to get inside the cell, and there's different ways of doing this. But so the next step is to get inside the cell. Then they have to uncode, the genome has to be released, and so the capsid has to be opened. Then this is followed by transcription and translation. So proteins are being formed, viral proteins. The genome has to be replicated. New virus particles are assembled. And finally the virus has to leave the host cell again. So here is a picture in more detail. So you see here the steps. First there is the attachment, then the virus gets into the cell. There's uncoating, so the capsid is being opened and the genome is being released. And this genome is being released is used for several things. It's used for transcription and translation in this pathway, and it's used as a template for genome replication. And then the virus particles are assembled and they are released. So like I said, every virus has its own replication mechanism, but if you follow some general rules, and you can have an idea on how probably replication will work. So if we go through this step by step, so these are the first few steps. So we have viral recognition of the host cells and binding of the host cells with viral attachment proteins. Now, if you remember the picture I showed you of hepatitis B, you saw, for example, the glycoproteins on the outside of the envelope, and those proteins are used then to do cause the attachment to the host cell. Now then there's entry, and entry can go in different ways, and generally you could say that the non-enveloped virus enter the cell by endocytosis or viropexis. And you might wonder what is the difference between endocytosis and viropexis, and basically this is a very similar mechanism. So we know endocytosis, but endocytosis is basically stimulated by, this is a host mechanism. So this is a mechanism that the cell has to bring things inside, and this is used when viruses are endocytosed. Viropexis is a system very much similar. However, in this case, this is induced by the virus itself. So this is, you could see this as virus-induced endocytosis. Viropexis is, for example, typical for the picorna viruses, of which hepatitis A is one. Now, the enveloped viruses, they mostly enter by fusion, and that is because their envelope is very similar to the host cell. So basically, the envelope of the host cell can melt together or fuse with the host membrane, and in that way, the viral capsid is being released into the cell. So after this viral capsid is being released into the cell, either by fusion or by endocytosis or viropexis, the next step is that it has to be opened. And the goal of this uncoating is that the genome is being released to the site of replication. And this site of replication can be the cytoplasm. For most RNA viruses, replication happens in the cytoplasm. So what you see there is that the capsid is often opened already on the cell membrane. But this could also be DNA viruses are often replicated in the nucleus, so the host nucleus. So there you see that the capsid moves towards the nuclear membrane and is often encoded there. So the opening of the capsid and bringing the genome to the site of replication. Like I said, here you have, for example, the picorna viruses like hepatitis A. This is an RNA virus. So what you see there is that while the virus is entered, immediately the capsid is opened, and basically it looks like there's just an injection of the genome into the cell. So these are small variations, but what you should remember is that the capsid has to open, and either when the RNA is being replicated in the cytoplasm, this happens very early on, so near the cellular membrane. If you are dealing with a DNA virus that is replicated inside the nucleus, often the capsid is opened on the nuclear membrane. So then we have genome replication, and to have genome replication, basically what you need to do is you need to have mRNA. So we have all kinds of variations of genomes from viruses. It could be a single-stranded DNA, a double-stranded DNA like our own, a negative single-stranded RNA, a double-stranded RNA, or a positive single-stranded RNA. Now, mRNA has the same structure as positive single-stranded RNA. So if your virus has a positive single-stranded RNA genome, this genome can directly be used by the host cell mechanism as an mRNA and immediately be transcribed by the host cell machinery. Now in all these other cases, mRNA has to be formed, and this can be done in different ways. For example, you could copy the negative single-stranded RNA, and when you copy it or you make it a complementary strand, you get positive single-stranded RNA. And then you, in that way, create mRNA. But this is a mechanism that doesn't happen inside the host cell because we don't deal with it in this way. So this means that you need viral RNA polymerases. Now, if you have double-stranded DNA, their mRNA can be made by the host cell machinery, because I mean that's what we do, right? We have a double-stranded DNA genome, and we make mRNA ourselves. So this genome can just be inserted into the cell, into the nucleus, and mRNA is formed by using the host enzymes. Now, single-stranded DNA from the virus is usually just complemented, so to form double-stranded DNA, and this double-stranded DNA can then be used by the host machinery to create mRNA. And then finally we have double-stranded RNA, and this double-stranded RNA can be used to copy mRNA, but again this has to be done by using viral enzymes. Now, the last group that I just want to briefly mention, but you don't need to remember that much about, is the retroviruses. Those are positive single-stranded RNA viruses like HIV. However, the funny thing about these viruses is that they are not used directly as mRNA, but they have a step in between in which viral enzymes are used to make from this single-stranded RNA a double-stranded DNA, and then make mRNA from there. And this part is then incorporated into the human genome, and that's why what we know about HIV, and that's why these viruses are called retroviruses. So basically they take a strange way from single-stranded RNA to double-stranded DNA back to single-stranded RNA. But this is an exception, and generally all the positive single-strand RNA viruses work like this and can be directly used by the host machinery to make proteins. So once the virus is replicated and translation and transcription has taken place, so viral proteins are made, then you have viral assembly and the release. So those are the two last steps. Now, viral assembly takes place in the cytoplasm. Again, you see here there is translation and transcription, proteins are being made, and this assembly can happen in different steps. I mean, we don't even know at this moment what happens for each virus, but we know that some viruses, first the capsid is being formed, and then finally through the opening the genome is injected into this empty capsid, what we call a provirus. But sometimes also the capsid is being built around the genome. Now, those are details. You just need to know that it can be different, but you don't need to know how that happens for each virus. What we do know more about, and what is a bit more important, is how the virus leaves the cell. Now, if we deal with an envelope virus, generically, these viruses are released from the cell by budding. And what you see here is that a capsid with a genome is being put inside the cell membrane, and then the cell membrane is budded off and forms the envelope around it. And I told you in the beginning that why they can enter by fusion is because the envelope really resembles the host cell membrane. Well, the reason why that is, you see here, because it's actually built off of a host cell membrane. And the only thing that happens is that what the virus does is it expresses its own viral glycoproteins on the cell membrane, and that's why you see them here. So the viral glycoproteins are expressed outside of the host cell membrane, and then it's budded off. You have a question? No? Okay. Okay, so the envelope viruses mostly exit by budding. In very rare cases, but one of these exceptions is hepatitis B virus, the envelope is acquired inside the cell. So the envelope is already made in the endoplasmatic reticulum, and then the viruses exit the cell either by cell lysis or exocytosis. Most of the naked capsid viruses, so the ones without envelope, they exit by lysis, and examples of that are noro and rotavirus. Now you can see that when the cells exit by lysis, this automatically means death of the cell. So you can see why this is important to know, because it does damage. Hepatitis A virus, for example, leaves the cell by exocytosis. So here the cell stays intact and alive. I mean, it's being used not for its normal function, but for virus production, but it's still intact and there. And the same for the viruses that are used for budding. However, if you think back about our immunology part, you can see here that when these viral proteins are, being put by the virus on the outside of the host cell membrane, these are very nice markers for our immune system that something is wrong with this cell. So it's quite obvious when all of a sudden you have a human cell there with viral glycoproteins. So just to give you a more overview, so what are common features of most of the RNA viruses? Most of them replicate in the cytoplasm, so if you have to have an educated guess with an RNA virus, you can see that they would mostly replicate in the cytoplasm. The cells cannot replicate RNA genomes, so there's always viral RNA polymerases necessary. And the only ones that don't need this, so they always need an RNA polymerase, and most of them actually also need to carry that RNA polymerase with them. So all the viruses except for the positive single-stranded RNA virus need to carry this RNA polymerase with them. And just to show this to you again, I mean here, why don't you need to carry a viral polymerase inside your capsid? Yeah, exactly. So because the RNA can immediately be turned into a protein, as long as the protein encodes for a polymerase, it can always be made. With the other one, they need to first make this positive single-strand RNA, and they can't, well, these two, they can't do that with the host polymerases. So they need to bring a polymerase that they can use to make this single-stranded RNA, and from there they can use the host mechanisms again. Is that clear? Yeah. So again, so for RNA genome replication, there's always viral RNA polymerase is necessary, and all of the RNA viruses except the positive single-stranded RNA virus need to carry this RNA polymerase inside their capsid. The other thing is that RNA viruses are prone to mutation, so there's very often mutations happening in their genomes, and that makes it difficult for us as humans to deal with this. So an example is, for example, influenza viruses, and we know that influenza virus is changing from year to year to year, and it's hard for us to build up immunity or to make vaccines. Well, so that is because RNA viruses are prone to mutation, so they are much more unstable than DNA viruses. So they change much more frequently, and it's harder to build up immunity and make vaccines. So if we then look at the positive single-stranded RNA virus, and what do they have in common? So they can be translated or used as a template because of their positive polarity. They can act directly as an mRNA and be translated directly to viral enzymes. So even if you would have a naked genome, so without the capsid, and if this happens to come into a cell, it is infectious by itself because it can use host enzymes. And here are some examples of this: hepatitis A, hepatitis C, hepatitis E, and norovirus. So many of the viruses that we're discussing in this block fall into this category. Then you have some common features of negative stranded RNA viruses. So this is antisense RNA packaged into a capsid. This naked RNA is not infectious because it needs the viral polymerase to make positive single-stranded RNA. It has to carry its own polymerase because there is no human RNA-dependent RNA polymerase. It doesn't exist. So the cellular location of gene expression and replication varies, but again, like I said before, if you have to have an educated guess, it would usually be in the cytoplasm. And this, so far, is all the negative RNA virus that we know thus far all carry an envelope. We don't really know why, but this is the case. So then if we use the double-stranded DNA viruses, now they have much more complex strategies for replication, but basically they follow the central dogma that we also know in human cells. So from DNA, mRNA is made and proteins are being made. And because this resembles the system that we use in our human cells so very much, this virus can use host enzymes or encode their own. They can be flexible in that way. So the DNA genomes generally replicate in the nucleus. Because the reason for that is because they use the host enzymes, and our own DNA replication happens inside the nucleus. And what it does is then it encodes viral, it expresses different viral genes, encodes viral proteins to manipulate the host cell and defenses in multiple ways. Like I showed you, for example, by expressing viral glycoproteins on the outside of the host cell. And there's different host evasion strategies, and an example of this is hepatitis B. Now, why am I focusing so much on this viral replication? And the reason for that is basically similar to what I showed you last week for the bacterial cells. And you see again this picture that we started with, and in red actually you see different antiviral compounds. So this whole process that is happening here has to be stopped, and different antiviral antiviral compounds can be used for that. And the reason why we can't just say, well, let's stop DNA replication, that is because this it uses our own host cells. So we have to make sure that these antiviral compounds don't work against our own cells. So we have to be very specific against these different steps. So uncoating is of course something that doesn't happen in our own cells. So this is something that could be targeted. We will go much more into detail into this in block 2.4 next year, but I just want you to know why we're focusing now on this replication, and that is because this is important. If you understand replication, you also understand how we can use antivirals. Now, told you before that because of their nature, and meaning that they use host cells, they are usually always considered to be pathogens. And how does this happen? Like with bacterial pathogenesis, there are several steps. Again, we have acquisition, so entry into the body, initiation of the infection at the primary site, and activation of innate proteins. So the viruses attaches and infects. Then there's usually an incubation period when the virus is amplified. And what you see there is, you can imagine how this works, because it's not the second that a virus infects a cell that we start to become sick, because the virus needs time to take over the cell and to turn the host machinery into its own. And even if only one cell is infected, it doesn't really mean that therefore we start noticing something. However, this usually goes quite fast, and this we will discuss in the next case when we talk about different sources of diarrhea. And you will see that diarrhea caused by a norovirus or rotavirus occurs, or the symptoms occur much faster than, for example, you would have a Campylobacter or a Vibrio. So there's replication in the target mechanism, and then we start to get immune responses. And often it's even the immune responses that cause us to be more sick than the viral replication by itself. Because like I told you, for example, for hepatitis A, the virus doesn't kill the cells. So it takes over some of the mechanisms, but the most damage is actually being done by the immune system trying to clear up the infected cells. And then in the end, we can have resolution, so the clear up of the virus by the immune system, or this could be a persistent or chronic infection. So what are the outcomes? Like I said, I mean, we could have a failed infection. It could be that the virus is trying to infect us, but it doesn't fully work, or before it starts spreading, the infected cells are already cleared out by our immune system, so that's an abortive infection. We could have cell death, biolytic infection, so if the cells lyse at the end of the viral replication, then the result is cell death. Or we can have replication without cell death. So this could be a chronic infection or a non-lytic productive infection, a latent infection. And a latent infection is basically an infection where no viruses are being formed. So the genome is there, and but no viral proteins are being formed yet. Now this is of course a quite complex mechanism, and people have been trying to find out why it works this way and what is the trigger for a virus to become go from latency into a productive infection again. Now for some viruses we know this, for example a herpes virus that give you the cold sores on your mouth, for example. Now what we know about this virus is that it becomes latent, so the genome is present in your cells, and only when your immune system becomes weakened this virus is being triggered into becoming productive. And then once that is being suppressed either by medication and the combination with the immune system, then it goes back into latency. And finally we can have a recurrent or transforming infection. So here you see an overview of this. So we have the different types of infections. Here an abortive or a cytolytic. Cytolytic is another word for a lytic or a virus, an active virus production that causes lysis of the cell. So like I said here, if there's an abortive infection, no virus is being produced and obviously nothing happens to the host cell. If you have a cytolytic infection, then there is virus produced, the cell is lysed in the end, and you have cell death. And then you can have persistent infection where it's being the virus is being produced but at a very low grade, and actually the immune system is being evaded, and so the cell is there and virus is being produced. And then a latent infection where no virus is being produced and there's no effect to the cell, and a transforming infection, and usually you have immortalization of the cell, and this is for example for those viruses that cause cancer. So the cells are being turned into cancer cells and then live on quite a long time. So those are actually the ones that we're focusing on most in this blog. So again, a bit more of an overview of the different patterns of infection. Like I said here, for example, for rotavirus, what you see here is just an acute infection. So it's a rapid onset, it's short-lived, and the virus is being cleared. And these viruses are all, like, for example, also hepatitis A are called hit and run viruses. So basically, before the immune system clears the infection, millions and millions of viral copies are being produced and being shed, and the next person will be infected. So you have a very short onset, and then it's cleared by the immune system. Now you can also have a persistent infection, so these are very, very long-term infections. So the infectious agent remains in the body, and it can be with or without symptoms. You have here latent reactivating infections. That's what I just told you about the herpes virus. So you have, you see a flare-up of the virus production, then it goes down again, and then when the immune system weakens, it can come back, and this can happen many, many times in the life cycle. And then finally, we have viruses, a slow viral infection, for example, HIV virus, where you have an acute viral infection at the start, and then the virus sort of lays low for a long time, and finally you have a final flare-up of the viral infection. Finally, I just mentioned something about viruses that can transform cells and cause cancer. There is different examples here. The one that we focus on in this block is hepatitis C. So this is in fact, for those of you who have paid attention, not a DNA but an RNA virus, and it can cause carcinomas. So how are viruses transmitted? This can be in many different ways. It can be through the fecal-oral route. It can be droplets, like you see here, which happens for all of the respiratory viruses. We can have sexual transmission, blood to blood, or vector mediated, for example by a mosquito or by ticks. And the diagnosis of viral infections has been done for years by viral culture. The reason is that, I showed you in the beginning that the only way that we can see viruses is by using electron microscopy. But electron microscopy is not that easy to do, and not many places have electron microscopy facilities. So this is not something that's commonly used for diagnosis. So what was being done was viral culture, and the way that happens is that what you see here, so human or animal cells are being cultivated. Is actually an example of monkey kidney cells, and these monkey kidney cells are being grown in monolayer in flasks. So you see here that all these cells, they're happily growing, and then they are infected with a virus, for example hepatitis A virus, and this virus starts to infect these cells, and cells can eventually will die from this, and then you start seeing that more and more cells are dead, like you see here. And finally, so what you see is that something is happening to the cell layer. So this is an indirect way of seeing that something is happening. So we have a lot of virus production here, and finally you see death of the cell, and this you can see, since the host cells can be easily seen by microscope. So you can study this in the lab. Nonetheless, this is not used so commonly anymore, and the reason for that is that not all of the viruses that we know can be cultured. For example, norovirus, we still do not have a way to culture this virus effectively. But also in the viruses that can be cultured, culture takes quite a long time, and long meaning between 1 and 2 weeks. So if you have symptoms of an acute virus, you don't want to wait for 2 weeks to find out if you actually do have it and which one it is. So what we do nowadays is we do infection serology, including rapid antigen tests. And these tests are, you would recognize, for example, they are the tests similar to a pregnancy test. So what you would do, for example, is you would take a drop of blood, or you take a throat swab, and you put a droplet of that in fluid and put it on this little test, and then in 2 minutes you get a band if it's positive, or no band if it's negative. So these are the rapid antigen tests, and they are, for example, available for noro and rotavirus. So this way in the laboratory we can see within a few minutes whether someone, or for example a child, has a rotavirus infection. But what is being used more and more now is molecular diagnostics, and here we detect the genome of the viruses by a PCR or by reverse transcriptase PCR, and detect this in a very sensitive way. Because these rapid antigen tests might be very rapid, and that's an advantage, but they're not very sensitive, so they do not detect low amounts of virus. So for treatment, there are no general antiviral drugs. So there's no drugs that stops replication of each virus, or entry of each virus, or stops uncoating of every virus. And this is logical again, because the reason is that they are all doing this in such different ways, and so that's why they can at most target groups of viruses, or sometimes also even single viruses. Sadly, most virus infections cannot be treated yet, but unlimited drugs are available for herpes viruses, HIV, influenza, and the hepatitis viruses. But a lot of these treatments exist actually of our boost of the immune system. So we give, for example, interferons, and I hope you all know now what interferons do in our immune system, so they can boost the antiviral mechanisms. So since treatment is not such a really good option, prevention with viral infections is key, And a very good way that we have for the strategy that we have for that is vaccination. Again, this is something that we have already discussed in this blog before. And for example, good hygienic practices. Sometimes we can give prophylactic treatment. For example, this can happen for influenza. What we have there is compounds that prevent entry, or the first steps of the viral pathogenesis. But if you want to cure someone by preventing entry, you should better start giving this treatment before someone gets infected. So basically what you do is you block that the virus can get into the cell, and in that way you prevent that someone gets sick. But again, hygienic measures and vaccination are the key strategies that we have. So to summarize this first part, I hope that you remember the general features of several viruses, and mainly the general properties of replication, that there's different routes of transmission, that treatment is limited, and therefore prevention is key in limiting viral infections. So the last part, I want to focus a bit more on gastroenteritis, and that is mainly for the next case. So what is gastroenteritis? And this is a combination of symptoms like vomiting and diarrhea. It could be non-inflammatory or inflammatory. Go a bit more in detail about that. So if we have diarrhea, what does diarrhea really mean? Well, it means that you have a very frequent and non-firm stools. So frequent, well, usually we say that the normal frequency of producing feces is about 3 times a week to 3 times a day. So if it's more than that, then or there's a sudden change in frequency and consistency of stool compared to a person's normal habit, you talk about diarrhea. And usually this means that since the consistency is different, it doesn't mean that it's more dry, but it means that it contains more water than usual. There's different ways how diarrhea, or different mechanisms of diarrhea. We call about exudative diarrhea, and usually what you have here is there's ulcerations in the colon, and that way water absorption does not happen very effectively anymore, and usually caused by an infection. Then you have osmotic diarrhea. That's if you would be fasting, you have less than 500 grams of defecation, and usually this is caused by non-absorbable intraluminal substances. So that's why it's called osmotic diarrhea. So basically a lot of water is being in the upper GI tract is being extracted into the gut. And then you have secretory diarrhea, more than 500 grams of defecation during fasting, and there's absorption and or secretion of electrolytes. So again, you're losing a lot of water and electrolytes there. So if we then focus on the infectious causes of diarrhea, so we have acute diarrhea, and it could be the watery one, which is caused by toxins, viruses, or bacteria. Now, the toxins I talked about in the previous lecture, which could be, for example, being produced by Bacillus cereus or Staphylococcus aureus. And then there's different viruses or bacteria that cause damage to our intestinal lining and cause watery diarrhea. And then there could be the heavily inflamed cases, which are usually caused by bacteria and parasites, and they are really causing an infection and invasion of our deeper, that goes beyond just the epithelial cells, but there's really invasion, and then we talk about dysentery. So since we're talking today mostly about the viruses a bit more in detail, there's different RNA and DNA viruses that can cause diarrhea. We have different picornaviridae, which are enterovirus or hepatitis A, caliciviridae, noro and astrovirus, and the reoviridae, like rotavirus, and then some DNA virus like adeno. In red, again, the ones that we are discussing in this block. And especially for rota and norovirus, I want to tell you a little bit more about that. So this is rotavirus. This is a double-stranded RNA virus. This is the way it looks. So you see here what it has is that it has a double capsid. So this is not, so remember this is not an envelope. This is a double capsid, so no envelope. And you see here the double capsid here in blue and in yellow. So this is belongs to the Virology, and what it stands for, respiratory enteric orphan viruses. So this is a bit of an odd one in the respiratory section. This is mainly called by viruses that were first found. There are different groups of rotaviruses. Some infect only animals, others infect also humans, and there's no envelope but a double capsid. The reason why I'm really saying this so clearly that it doesn't have an envelope is that because it doesn't have an envelope, it is stable in the gastrointestinal tract. And this feels a bit like counterintuitive, because you think, well, when it has an envelope it should be extra protected, and that's why it's stable, but in fact this is not the case, and an envelope is usually a more unstable part. Since these viruses are transmitted through the oral fecal route, it means that they have to go through the stomach. And in the stomach, a lot of glycoproteins are being inactivated. So what you see is that an envelope is usually destroyed in at the low stomach pH. So in the stomach, envelopes are usually destroyed. And that's why pretty much all the viruses that cause gastroenteritis do not have an envelope. So that's also rotavirus, does not have an envelope but a double shell capsid, and is therefore stable in the GI tract. Now the genome consists of 11 distinct double-stranded RNA segments, and that encode 11 viral proteins. And the way this works is usually one major protein is being formed and is then being cleaved in small parts. And some of them that are brought is the RNA polymerase. Of course, that is needed to make from this double-stranded DNA genome the mRNA, but also some surface proteins. This is a rough overview of this virus replication. Now, the virus is an envelope or an virus without an envelope. So, as predicted, this enters the cell by endocytosis. It is an RNA virus, so it replicates in the cytoplasm. So the capsid, or the uncoding of the capsid, happens quite close to the cell wall, or the cell membrane, I mean. Then the genome is being released. It is being replicated by its own RNA polymerase. And when mRNA is formed, the cell machinery in the cytoplasm is used to produce viral proteins. Then the genomic RNA genome is being combined with the viral capsid proteins, and then is being released, like many of the non-envelope viruses, by lysis. So I hope that by the steps that we discussed before, that you could now predict that this would be happening. No, double-stranded RNA. Yeah.
But minus. But it's minus.
No, it's double-stranded. So you have single-stranded plus or minus, or double-stranded RNA.
The double-stranded uses the RNA polymerase, which belongs to
the virus.
Yeah.
Yes. Now, to give you one picture a bit more in detail, and to show you a bit the combination of how the immune system works together, or how the virus works around the immune system, I wanted to go through the pathogenesis of rotavirus with you a little bit in more detail. So what you see here is that you start with the virus, and we have here we have here antibodies, human antibodies, that are being directed against some of the surface proteins. And what these antibodies try to do, I hope you remember this is a neutralization. So basically what it tried to do is to cover the virus completely so it cannot attach to our epithelial cells. What it does is it tries to neutralize it, but when it cannot neutralize effectively, then the virus binds to our enterocytes. And so what happens is the virus attaches to the enterocytes, is being taken up by endocytosis, and is being used then, like I just said, it uses its own RNA polymerase and the host cell host cell machinery to make proteins and make more viruses. So what you see here is that you have some IgA. When the IgA is present in the enterocytes, again, it can already stop blocking things here inside the cell, but at the same time, when that doesn't work, there are symptoms that the cell is being infected. So you see that there are some T cells here that are producing cytokines, and they try to make sure that it is being noticed that the cell is being infected. So they try to inhibit viral replication. Now, if the viral replication is not stopped, this rotavirus starts producing proteins, and these proteins are then being used to help it spread. So what you see here is that and this is still a bit unknown, the rotavirus is then being spread from cell to cell, and you see that it's going here from different cells to each other. And finally, cells then are being lysed and more virus is being released. So again, I mean, not all the details are known, but you see here how there's a combination of our human immune system that is trying to combat these cells, and the virus is trying to work its way through this. Now, luckily, since our immune system is quite effective, what happens in rotavirus epidemiology is that every time that we get infected, the immune system combats the virus more effectively and quicker. And what you see here is this is the average age in months of, so when a baby is born, and here you see the probability of rotavirus infection. So in the first infection, when your first infection happens at a later age, but I mean this is only within the first 2 years, you see that there's high probability of getting infected, but the more times that you get infected, or the more times you get in contact with the virus, the chance of getting infected is lower because our immune system is ready for it. Now, so that is a key message, but the second key message is that this happens usually at a very early age. And rotavirus is typical for being the main reason for diarrhea in children. And this develops a bit, or this is both for developed and undeveloped countries. So you see that rotavirus by itself is the main cause of gastroenteritis in children. The rest of the causes vary a bit. In undeveloped countries, there is a lot of bacteria that cause gastroenteritis. In developed countries, the part of bacterial is only rather small. Now, since rotavirus happens everywhere, sadly, the people that die from rotavirus, or the kids that die, are mainly in the developing countries. And the main reason why people die from rotavirus is that they lose a lot of fluids and there's dehydration. So it is the most important cause of gastroenteritis. In developed countries, you see that 50% of all hospitalized diarrhea cases with severe dehydration, and that is mainly with children, are due to rotavirus. In developing countries, there's almost 1 million deaths per year due to rotavirus. And they infect the enterocytes of the intestinal villi. This is a lytic infection, so the cells die, and the infection decreases water absorption and causes loss of ions, so watery diarrhea.
The cholera, is it belongs to bacterial infection or?
Yeah. And that's one of the reasons why that is in developing countries a much bigger part. So here you have an idea on how many viral particles are being released per gram feces. Now 10 to the 10, I mean, it is billions basically of copies of viral particles per gram feces. And it spreads via the fecal oral route. Now I'll come back to this in a second. So the infection is usually self-limiting. Like I said, it's generally seen as a hit-and-run virus. Dehydration is the main contributor to mortality, and secondary malabsorption could be of lactose and fat, and chronic diarrhea is possible, but not that common. And there's no antiviral therapy, only supportive therapy, so that is by rehydrating the children. And recently some vaccines are become available, and there's two of them. I just show you some characteristics of those. They are both live viruses. One is a live weakened virus, one is a live virus that is has some RNA reassortment. This is where there's a shedding of the attenuated strain, yes or no, and then basically you can spread this immunity around. In the Netherlands, this vaccine is not commonly given. There are some other countries, like Belgium, for example, that are experimenting with giving small children these viruses. And this is not because in Belgium this virus is all of a sudden so much more dangerous, but you can imagine that if you go to a daycare center and all of the kids are having rotavirus infections, all the parents have to stay home, and there's a huge economic burden of these rotavirus infections in developed countries. So again, the efficacy of the both vaccines is 80%. Now that's not 100%, but what happens in the kids that are vaccinated and still get sick is that the disease is generally milder. So this is, again, like I said, in the Netherlands there's a discussion at the moment whether or not we should add this vaccine to our national program. And the positive, of course, is that you have reduction of disease. The benefit or the disadvantage is obviously that it costs money, and that there's no mortality. And it's mainly of economical value. Now then, a little bit more about the noroviruses. The noroviruses belong to the Calliciviridae. And are named, so they are called Norwalk-like viruses or noroviruses. And they're called after a big gastroenteritis outbreak in Norwalk, Ohio. Like rotavirus, these are non-enveloped viruses, so naked viruses, and because they are naked, they are stable and they survive transport through the GI tract. And they have one single-stranded positive RNA genome that encodes 3 proteins, among which the polymerase and a capsid protein. The replication mechanism is more or less like the picornaviruses, so like hepatitis A, so which you have studied a bit for today. Replication most likely in the cytoplasm, and what happens again is the virus-positive RNA is turned into a virus-negative strand RNA, and from that negative strand the positive RNA genomes are being replicated, and this happens through the viral RNA polymerase. Noroviruses are the main cause of gastroenteritis in adults, and it causes a lot of outbreaks in centers where there's a lot of people eating from the same food sources or sharing the same toilet. So you see that commonly in schools, daycare centers, unfortunately hospitals, but also cruise ships are actually quite well known for having norovirus outbreaks. And the reason why this is so contagious is that the infectious dose is very low, so 10 viral particles. And like I showed you for rotavirus, that in the stool of someone that is infected you can find 10 to the 10 viral particles per gram. You can imagine it is similar for norovirus. So you have 10 to the 10 viral particles per gram, but only 10 viral particles can make your neighbor sick. So you can imagine how easily this spreads, and you have to wash your hands extremely well. Norovirus causes nausea, vomiting and diarrhea, and it occurs rather quickly, so within 2 hours to 2 days. and spreads via the fecal oral route. So, and the source is usually contaminated water and food related. So you can see also salads, oysters, water. These are all things that are have to do with contaminated water. Again, the infection is self-limiting, and there is treatment that is usually not necessary. There is also no antiviral treatment, and like I said before, until a few years ago we couldn't even culture the virus, so it was very hard to determine whether we were dealing with norovirus. So prevention is really through improved hygiene. And just to give you a brief overview of Rota, Noro, and hepatitis A viruses, so those viruses that can cause gastroenteritis, you see here that we're dealing with RNA viruses, one double-stranded, two positive-stranded RNA viruses. They all don't have an envelope, so they're stable in the GI tract. Rota and Noro cause lytic infections, whereas hepatitis A does not cause lytic infections. There's no latency of the virus. The transmission is mainly by fecal oral route, and the infectious dose is very low, so 10 to, in this case, maybe 100 viral particles, so highly contagious. And that was what I want to tell you this morning. So, are there any questions? No? Okay, well, thank you for your attention then.